moving on to chapter 6, which is related in some ways to chapter 5. There's a lot of chapter 5, which is about sex determination, how that relies sort of on chromosomal content of the cell. We're really going to focus on, in chapter 6, on basically chromosomes and how chromosomes vary in their number and arrangement. And we're going to start today by just talking about numbers of chromosomes that exist in cells. So we're going to talk about how they vary in number and what are the implications of something that's called aneuploidy. Now, we already encountered this word aneuploidy last week. Does anybody remember what that means? Yes, exactly. It basically refers to for a single chromosome receiving any number other than 2 for diploid organism, right? So Caitlin's right. We talked about a few of the X chromosome related aneuploid mutations. Right? We talked about Turner syndrome, in which case you only have one X chromosome and no homolog to it. And then we talked about 3 different syndromes that result from trisomy of sex chromosomes. It could be XYY, XXY, or XXX, right? Those are 3 different syndromes. So now we're going to apply that to sort of other chromosomes in the nucleus, so the autosomes, okay? I thought I remembered this, but we talked about it last week. I don't really like showing pictures of humans with genetic defects, and that's just because I'm not always sure that those people agree to be the poster children for whatever genetic disease they have. If you're interested in any of these symptoms, feel free to look them up on your own. How does variation in chromosome number even happen? When we talked about the cell cycle and how that fits in with the eukaryotic life cycle, one of the things that we discovered is you have a diploid condition, you have 2 sets of chromosomes. At some point, an organism will undergo meiosis, which will reduce those numbers of chromosomes by half, eventually producing gametes, which are haploid. And then when 2 eukaryotes love each other very much, the gametes get together, forming one cell called the zygote, restoring the diploid condition, okay? That isn't always what happens. And just sort of to reiterate some things we talked about last week, we did mention non-disjunction as being sort of the leading cause of aneuploidy for sex chromosomes. And that's true of autosomes as well. And non-disjunction is simply the failure of chromosomes to migrate properly during meiosis. For any set of chromosomes, right, we can have a failure to migrate properly either in the first part of meiosis, that first division. So in that case, the homologs end up in the same cell, right, which ends up being a diploid cell, right? And then the other cell doesn't receive any copies of that chromosome, and ultimately you end up producing gametes that either have 3 copies, which is called trisomic. When you have 3 copies of an autosome, you call it a trisomic gamete, right? Or we'll end up having none, right? So we can, I should mention that the trisomy results from taking these diploid gametes, have 2 sets of chromosomes, and combining them with a normal haploid gamete to result in 3 chromosomes, right? Or if we unite that haploid gamete with one of these empty gametes that doesn't have a copy of that chromosome, you end up with just 1, which is something that we call monosomy. Does that make sense to everybody? I probably could have explained it a little better. I feel like that is always true. The other thing that can happen is we can have a failure of separation in the second division of meiosis, which is essentially like mitosis, because we're separating sister chromatids. And so the sister chromatids migrate together. In the second division, we're going to produce 1 of 4 gametes, which is diploid, one of them which has no copies of the chromosome at all, right? And again, when we combine that with a normal haploid gamete, we're going to end up with some of our offspring being diploid, right, disomic or normal, and then we'll have some of them be trisomic, having 3 copies, and some of them being monosomic, which means that they have only 1. Does that make sense to everybody? I feel like it'd be cool if they kind of switched the chapters around. So we talked about this with autosomes before we talked about the sex chromosomes, because it's just much simpler with autosomes. Anyway, how does this actually affect the organisms that inherit aneuploid sets of chromosomes? It depends a little bit on the organism. Oftentimes, we have a condition what's called haploinsufficiency early on in development. And haploinsufficiency is just the opposite of haplosufficiency, right? If you are haploinsufficient, or if a chromosome or a gene is haploinsufficient, it means that one copy does not produce as much of a phenotype as two copies, right? And so the number of copies that you have of a particular gene will determine, to some extent, the phenotype of the particular organism, okay? And so since haploid organisms or monosomic organisms, right, will express the gene less, right, they'll end up with some differences in phenotype. Now this has the greatest effect for genes that are really, really important in development. It's really the first stages in complex multicellular eukaryotes that determine a lot about whether an embryo survives and forms all the sort of functional items that it needs. But that also varies a lot among different kinds of organisms. So some organisms can handle aneuploidy really, really quite well because their development has a tendency to be really simple. When I talk about that, I'm talking about things like plants. Plants have really, really simple development compared to other multicellular eukaryotes, right? Just when you think about it, right? If, just numbers of organs. Well, to begin with, how many organs does a plant usually have? Like 3, right? It's got leaves, stems, and roots. Really pretty simple, right? I don't even know how many organs I have, but I know it's more than 3. Insects have more than 3. Complex eukaryotic animals, metazoans definitely have lots and lots of organs. And not only that, but we have to have the right number, right? A plant can basically have any number of leaves, and it's just going to be fine. They grow, they fall off. It doesn't really matter so much the number, right? If you have 2 hearts, you're in trouble, right? And so organisms with complex development, like mammals and insects, right? Most of the animals, right, really, really suffer oftentimes from aneuploidy in a way that more simple, simply developing multicellular eukaryotes don't. Right? So when we see aneuploidy, it generally is not that damaging in organisms like plants and fungi, and it can be super damaging in organisms like animals. Few terms to deal with, and some of this should be probably review at this point. When we talk about organisms that have the expected number of chromosomes, those are what we call euploid. You guys remember from previous science classes, what does U mean? Whenever you see EU together, it means true or good, right? It's or like I guess what you would call like the expected or the normal number. Okay, aneuploid means not euploid, right? And so that means not the expected number. Keep in mind that aneuploidy only refers to copies of a single chromosome. Okay, so you can be aneuploid for one chromosome but have euploid numbers for the rest of the chromosomes. If you are aneuploid, most likely if your euploid number is diploid, right? Normally you have 2 sets of chromosomes per cell. If you're aneuploid, you're most likely to have 1 or 3. And if you have 1 set of chromosomes in an aneuploid chromosome, that's called monosomy. Literally means one body, right? Or if you have 3 copies, you have trisomy. Okay. This is really different from something we're going to focus on much more in the second half of today's lecture, which is called polyploidy. Polyploidy is a doubling or tripling or quadrupling of essentially all of the chromosomes in the cell. So you have a non-disjunction event that basically affects every chromosome in the cell, and that's usually because you have a failure for the centriole to form at all. Right? So with division, the chromosomes don't migrate, and all of them end up in a gamete rather than haploid. Okay? When an organism has 3 sets of chromosomes, we call that triploid, and when it's got 4, we call it tetraploid. And as you can imagine, we could just keep going up from there. We'll talk a little bit later about some organisms which are octoploid. They have 8 sets of chromosomes in each cell. I'm just going to tell you the first time I ever heard about this, it made me feel super interested all of a sudden in genetics. And it sort of, I think, eventually led to, partially led to my career as an evolutionary botanist. Because polyploidy's got all kinds of cool and interesting genetic sort of implications. We'll talk about those later on. Okay, but everybody fine with the terms? Okay. If you're not later on and I'm saying something you don't understand, go ahead and stop me, okay? We rolled through material at a very high pace last week, and we don't necessarily need to. If you feel lost, you can go ahead and stop. All right, so what about people? Polyploidy never happens in people. There are no people that have 3 sets of chromosomes in every cell. There are no people that have 4, right? We are too developmentally complex for that to happen. And in fact, aneuploidy is also really, really rare because of, again, the developmental complexity of embryogenesis. If we have cells that have any aneuploidy at all in them, so let's say we have a zygote that ends up with trisomy of 1 chromosome or monosomy of chromosome, 20% of the time, those all abort spontaneously. So they don't even sort of get to sort of a fetal level of development. Almost all of the ones that exist demonstrate aneuploidy, or of those that do demonstrate aneuploidy, 30% are almost trisomic for at least 1 chromosome. And so this is something which doesn't occur uncommonly, but we just don't often see the results, right? So human pregnancies often result, well, not often, but 6% of the time, result in aneuploidy, and that's mostly from syngamy between aneuploid gametes, but most of those don't survive. Of the ones that do survive, we already spent some time talking about aneuploidy at the X chromosome, which results in Turner syndrome, Klinefelter syndrome, that triple X syndrome, and as well as XYY syndrome, right? And in some ways, the X chromosome has sort of got a built-in buffer against dosage effects anyway, right? Because of lionization. That's sort of a normal thing. So when you think about it, for the X chromosome, males are, we say hemizygous, but they're also aneuploid. You only have 1 copy of X rather than 2. Right? And so we have a built-in system to sort of deal with those dosage effects. We don't really with all of the autosomes. There are really only 3 human chromosomes that we have documented aneuploidy in, where an individual survives embryogenesis, essentially. And that can happen at chromosome 13, which results in something called a Patau syndrome, at chromosome 18, which results in Edwards syndrome, and also in chromosome 21. And trisomy of chromosome 21 is called Down syndrome. We're going to talk about each of these 3 individually. I will point out that Patau syndrome and Edwards syndrome are largely lethal. It's very, very rare that an embryo survives to birth and through childhood with these with aneuploidy at these particular chromosomes. Patau syndrome results from trisomy of chromosome 13, which means that one parent has a diploid gamete, so they're donating 2 copies of that chromosome rather than 1. So you end up with 3 copies. This occurs in 1 to 10,000 to 20,000 live births. Question?
And these are autosomal?
Yes, these are autosomal, right? So all the chromosomes that have numbers are autosomes, and the chromosome that doesn't have a number is X, right? So that's the sex chromosome, right? So these are all autosomes. They have nothing to do with it. So this occurs very rarely, 1 in 10,000 to 2,000 live births, right? Which really means that most of the pregnancies that produce a triploid zygote for chromosome 18 don't survive to that point. This has complete penetrance, which means that if you have 3 sets of chromosomes for trisomy 18, you're going to exhibit every bit of the symptoms that are, well, you're going to exhibit symptoms. You'll never be born without any symptoms, right? Expressivity is variable, which means that the degree to which you express symptoms is going to vary. Right? You might have some of the symptoms, you might have all of them, but you'll at least have some. There's a lot of symptoms that come along with this that are really the result of sort of errors in development. So cognitive and motor disability occurs in almost all individuals' live births that occur with Patau syndrome. Microcephaly is also common. Microcephaly is when the skull fails to develop fully and it ends up developing much smaller than is normal, and of course that impedes brain development. It's related to actually some of this cognitive and motor disability. When you have microcephaly, it's also often to have vision defects, right? Because there's not room in your orbital sockets to form functional eyes with functional nerves. Polydactyly is not uncommon. Can anybody guess what that is just by the word?
Extra finger.
Oh, you guys were quick on that. Yeah, extra fingers, right? I don't know about any of you guys, but I dabble around on guitar, and I've often wished that I had a few extra fingers on this hand. Not this one so much, but like 6 or 7 fingers. Also, this sometimes results in something called cyclopia. Cyclopia is a failure to basically develop the hemispheric structure in the skull. And so one of the big symptoms of this is people with cyclopia, or most often like embryos that are not viable with cyclopia, end up developing essentially one eye and one eye socket that's right in the middle, like a cyclops. They often have something called ankylosing, which is where the organs will actually form outside of the body to some extent, right? So you'll have sort of a membrane that passes through the navel, and on the outside that will be portions of usually the alimentary canal, like the large and small intestines. Cleft palate is fairly common, and then there are genital kidney defects and heart defects, which arise from this. So you can imagine that this is something that is not, people do not grow to reproductive age if they have trisomy of chromosome 13. Edwards syndrome is when you have trisomy of chromosome 18, again, largely lethal. It occurs in 1 in 1,000 live births. It's also got complete penetrance. So if you have these 3 chromosomes, you will exhibit symptoms, right, with variable expressivity. So you might not experience all of the symptoms, but you'll definitely experience some of them. Most live births don't survive for even 1 year without some heavy intervention in the form of usually surgery and like major surgeries. Often results again in cognitive and motor disability, microcephaly, something called hydrocephaly. Hydrocephaly is the production of too much spinal fluid in the brain, and that puts pressure on, brain tissues and causes those to malfunction as well. Arthrogyropsis, which is, let me look really quickly. Oh, I remember. I had to study because I don't know how humans work. Arthrogyropsis is when you basically have a shortening of the muscles, and what it does is it locks your joints in a particular position, right, so that your joints end up not flexible, right? So if you have, for example, a shortening of the bicep, right, it means that your arm can't stretch out, right? So it's just constantly locked up like that, and that happens to many of the joints. And finally, there's something which is called esophageal atresia. Esophageal atresia is when the esophagus forms poorly, so that it forms in more than one segment. So you might have a segment that occurs at the top, but this just sort of ends in a fistula, right? And then below that, you'll have the rest of your esophagus, which isn't connected, right? And so when that happens, it takes surgery to connect those up, or if a child doesn't get that, they have to get a feeding tube, or they'll starve very, very quickly. So this is, again, super dangerous, right? We don't tolerate aneuploidy very well, okay? The only aneuploidy that people can tolerate is something which is called trisomy of chromosome 21, and this occurs in 1 in 800 live births. This is actually fairly common, and the life expectancy of this is less than average, but it is a full life expectancy for the most part, like 50 to 60 years. Again, penetrance is complete. You aren't born without Down syndrome without experiencing some of the symptoms of it, right? But it will vary in the degree to what you express them. So people with Down syndrome often experience cognitive disability, and psychomotor disability just means it's harder to sort of move in a coordinated fashion. Often they experience heart defects, bone shortening, which results in sort of a characteristic appearance. People with Down syndrome tend to be a little bit shorter than their parents are. Low muscle tone, so they don't form like very much muscle very easily. An epicanthic fold is a fold that occurs next to your eye, right? So this gives sort of an almond-shaped appearance to the eyes. Sometimes the tongue is a little bit larger, so it sticks out of the mouth. And unfortunately, there's also a super high incidence of leukemia, especially childhood leukemia in kids with Down syndrome. And like I said, I normally don't show pictures of people who have these genetic diseases because I don't know with their permission, but in fact, I know a kid with Down syndrome really well. I talked to her. I've got some permission to show a little bit about her story. And this is because, I'm going to talk about other things first. I forgot. We'll get to that in a second. One of the causes of this kind of aneuploidy, and I think this is probably true for all of the aneuploidies, is an increase in age of the mother. As you guys probably know, human meiosis in females is usually arrested at metaphase 1, right? And it doesn't continue till later on. It's kind of interesting. So like every egg cell that each one of us developed from existed in your mother as an embryo, right? Your mother had that egg cell when she was an embryo. So really my first cell is from 1950, right? Your first cells are probably from the 70s, right? Which is weird when you think about it. They get arrested and then they'll just continue with meiosis later on. But what that means is that the older that oocyte is, right, the less likely it is to have properly functioning centrosomes, which results in a failure of kinetochores to link up with microtubules, right, and non-disjunction. Okay, so it's really common for older women. So this is basically just Down syndrome per 1,000 births, and you can see as the maternal age increases quite a bit. Okay, one of the things that I think is understudied is the paternal contribution to Down syndrome, and that's simply because even though sperm cells are usually just made like throughout a person's life and like in insane numbers that are not even necessary when you really think about it. They are usually made like on the spot, like really, really recently. However, older males do have higher probabilities of non-disjunction, right? Because they have older cell lines that has sort of accumulated mutations over time, right? And in fact, most mutations that are passed on in autosomes originate in males because you just have many more replication events basically from egg cell to egg cell in male cell line. One of the things that actually turns out to be sort of fortunate for people with Down syndrome, especially because they have such a high incidence of leukemia, is they have a lower incidence of metastasis, right? So they don't really form solid tumors very easily. So when they have those cells that are sort of growing out of control, they are often not vascularized, right? So they don't have sort of this vascular ingrowth, which results in the tumor not growing very, very quickly, which is sort of fortunate for them. So getting back to my niece, I got too excited because we're buddies. This is my niece, Anya. This is her, and that's me. This is from 2019, but she has Down syndrome, and it's kind of a really interesting kid. And one of the cool things about Anya is her mother's a filmmaker. And so when she was born, she really changed sort of her focus in how she was making films to spend much more time talking about sort of kids with Down syndrome, and she's become really involved in that. And she won an award for a little film that she made about Anya. I figure I'd show it to you because it's adorable. And it makes like the person seem like much less of a specimen and more of like a person who's just got some different genetic. I just washed this upstairs. Give me just a second.
Listen, that's crying every time. Who's red?
I really too, man. You gonna poop?
No.
I'm Alex. This is Will.
That's my brother.
And Max.
I don't always get to do push-ups all the time.
In Irish mythology, Anya means the best-hearted woman that ever lived. Lucky in love and in money. If you're going to be lucky, it's best to be lucky in both. So it's 2014. I'm 37. A geriatric pregnancy. I know, sexy, right? It's my 12-week ultrasound appointment.
The baby is a little small, but looks great.
Oh, close. She has one more test result to grab, and she leaves the room. Trisomy 21, Down syndrome, genetic counseling. Only so much time. You must get these tests. If you choose to terminate, you have to do it soon. You're gaining too much weight. She will live with you your whole life. She doesn't have a nasal bone. She may be born with her heart on the outside of her body. The most dangerous place for a child with Down syndrome is in the womb. She will likely have a thyroid issue, diabetes, or leukemia. She arrived after 2 pushes. I mean, even the midwife was surprised. I was told she wouldn't be able to lift her head for 6 months, and it was the first thing she did right after she peed on me. She works harder to do things than her typical peers. There are certain challenges in her life. She has low tone. On day 4 of her life, she had to go to her first feeding session. She has bilateral patellar instability, which means her kneecaps slide back and forth, and she falls a lot.
Yeah, piano plays fun too.
Will and I both come from very musical families, so music has always been a place of connection between the two of us. Music is about learning.
She was doing great. She was healthy until she got cancer. Leukemia is 10 to 15 times more common in children with Down syndrome. It felt impossible. Chemo has rotten side effects. Food tastes like metal. The roid rage is real. We chose to laugh. We chose to dance. We chose to sing. Her cancer went into remission. I want her to know a world of creativity. With focused attention, any limitations just appear to be hurdles she eventually soars over. I want her to know a world where no one can limit her. To me, having a child with Down syndrome means going farther than you knew you could.
You can do it, you can do it.
No.
Yes, you can.
What I wish for her is to know a world where she is not treated as other, where she is not compared to other children. More than anything, she's a kid. She's my kid. She's a child of this world, and she has the same needs and the same wants and the same desires as any other kid. Her brain and her body are just a little different, but the way she operates in the world is not.
I feel happy.
You feel happy?
You're happy too.
Happy too, you're right. She is on her very own singular path.
Right?
Yeah, I mean, how can I not show that to you? Like, when you talk about Down syndrome, like, she's a super. We'll play all day. Any questions about that? So we're gonna move on and start talking about people for a change, which is nice, and really focus on this sort of the sex thing that happens, which is polyploidy, which is no less important, and as it turns out, is way more common than a lot of people think. Remember, polyploidy is the spontaneous doubling or duplication of all of the chromosomes in a cell, right? So you're duplicating complete chromosomal sets. So we're going to spend some time talking about the developmental and evolutionary implications of polyploidy. And one of the things I think is fun about this is that the history of what we know about polyploidy really begins in the Soviet Union. Now, I don't think probably, not to pick on you, but Charles, maybe you and me remember the Soviet Union in this room. Maybe nobody else, right? This was a real thing when I was a kid. In 1984, they made this movie called Red Dawn, which is about the Soviet Union invading the United States, and a bold, plucky band of teenagers who fought them off, called themselves the Wolverines. It was pretty great. But this is something that was on our minds pretty much all the time. The Soviet Union was a large country that consists of many modern-day countries like Estonia, Latvia, Lithuania, and then we've got Kazakhstan, Uzbekistan, Turkmenistan, Azerbaijan, Georgia, Tajikistan, Kyrgyzstan, and largely influential in Mongolia. It used to be sort of really also largely influential in Eastern Europe. And for many years, the Soviet Union was sort of considered the opposite of sort of the Western bloc countries, the United States and our allies. And we participated in this thing that's called the Cold War, where we built lots and lots of nuclear weapons. They built lots and lots of nuclear weapons until we both had enough to blow the world up a lot of times over. But as it turns out, economically, this didn't work out as well for them as it worked out for us. And between really 1989 and 1991, it all sort of fell apart, ultimately with the dissolution of the Soviet Union into lots and lots of other countries, right, that we just sort of talked about. In any case, one of the earliest leaders of the Soviet Union was this guy named Joseph Stalin. If you haven't heard much about Stalin, he was bad. He's kind of a guy who killed a lot of people for not great reasons, was super big on imprisoning people in Siberia, which is sort of this big empty spot that exists in the middle of Russia. And he appointed one of his close political comrades Trofim Lysenko, who was an agrarian, sort of as the head of Soviet science. Now, one of the weird things about sort of the communist revolution in Russia is that it was an all or nothing sort of thing, right? So sort of the Soviet thinking basically led to all kinds of interpretations in other parts of human discourse that don't really have anything to do with economics, right? Like when you think about communism and economic system, science isn't, but at the same time, sort of communism really influenced the way that science was done. And Trofim Lysenko was all on board with this. He was an agrarian who simultaneously rejected Mendelian genetics and Darwinian evolution in favor of, really, we would recognize as Lamarckian evolution, which is this idea that organisms don't really necessarily differ much the way they're born, but they're shaped by their environment, right? And their environment can sort of, in some ways, determine how they pass their genes on to the next generation. Doesn't make a lot of sense given everything that we know about heredity, but he grabbed onto it in all but one way, right? He basically claimed that this was a Soviet idea, didn't give any credit to the French thinker Lamarck, who came up with it, because the French were the enemies. It's this weird, right? And it ultimately resulted in an enormous famine, because part of his agrarian system was, he said, well, you could take wheat and you could turn it into what they imagined was called winter wheat. So if you take the seeds and you basically let them sit at very, very cold temperatures for a number of years, then you could grow them during the winter in Russia, right? So while the snow is up, like you'd have these seeds and you could get an extra crop of wheat. Turns out you can't. Wheat just doesn't do that, even if you freeze the seeds. What happens is those seeds, right, they might germinate if it's wet enough, and then they'll die, right? So you lose a crop, and people starve on account of it. Nonetheless, despite the failure, he was in charge of Soviet science for a really long time. And he had appointed another agrarian, a junior agrarian, named Georgy Dmitry Karpachenko, to basically engineer the vegetable of the proletariat. Now, this was something that was an attempt at a hybrid between two really easily grown and commonly grown plants that are also closely related to each other: radish, which goes by the genus name Raphanus, and cabbage, which goes by the genus name Brassica. Okay, so we could call the hybrid, a rabbage or a caddish or Raphano brassica. And as it turns out, these two different genera are closely enough related, and they have a similar number of chromosomes. If you look at the number of chromosomes in the cell, right, they had 2 and 18. So even though Karpachenko was working for the Soviet sort of science institution, he'd been trained in science outside of the Soviet Union. So he knew about how Mendelian genetics works. He knew to look at chromosomes and things like that. You would do these, the idea behind, incidentally, of making a rabbage, right, or a caddish, was that a radish has got a super useful root, not much above ground. Cabbage has got a super useful shoot, right, all these delicious leaves above ground, not much below ground. And the idea was, what if we could have a plant that's got this radish at the base and a cabbage on top? We could use the whole thing, right, to have delicious cabbage radish base meals. This was something that was important to do. And Karpachenko was, in fact, able to get hybrids. And the hybrids were viable, or not viable, but they would grow, right? The problem is that they never produced seeds. And this is because they're sterile, right? Let me explain how this works then, right? When you have 9 chromosomes, right, so this has got a 2N number of 18. Every gamete is going to carry 9 radish chromosomes, and you combine that with 9 brassica chromosomes. As it turns out, the chromosomes themselves aren't similar enough that they match up very well during metaphase and prophase of meiosis 1. Right? They don't really match, and therefore they don't really segregate correctly. It's very, very rare that you end up producing any gamete that will actually function in this kind of plant, right? Because you don't have good buddies during homologous pairing of chromosomes. Is anybody confused about what I just said? Because as I'm listening to myself babble, it sounds stupid. If later on you're confused. So these were sterile. And it wasn't until, and he did this over and over again, right? Lots of pressure to get this to work. Until one time, he had one individual that ended up producing flowers and seeds, right? And when he went and looked at the tips of the growing roots, which is one of the ways to do a squash, a mitosis squash, he noticed that there were, instead of having 18 chromosomes, there were 36, right? And then when he started looking at meiotic squashes in the anthers, what he noticed is that there was 18 and 18, essentially, right? This resulted from a spontaneous doubling of chromosome number, right? So if you have 9 and 9, where the chromosomes don't match, and therefore you can't undo meiosis, right? That can be fixed by basically just doubling every chromosome, right? So if each chromosome is doubled, now it has a button, right? And so it can undergo meiosis correctly. So this spontaneous doubling of chromosomes restores fertility in otherwise sterile hybrids, right? Which is what Karpachenko discovered. And this is incredibly important, not only in agronomy, but it's important for our understanding of genetics and evolution of a lot of life on Earth. Here's the unfortunate thing, though, for Karpachenko, is that the cabbage that he made, or the radish that he made, had the root of a cabbage and the shoot of a radish. He never could get the other thing to work. And so he was sent to a gulag, a prison in Siberia, where he died at about the time that World War II started, right? This is the price of doing science badly in Russia, right? You don't just get fired, right? You have to go to prison, which is, how can anybody get anything done, right? Clearly it's a system that's not going to work. But this is an important discovery. And it was a discovery that he was able to publish before it was more or less executed by work and starvation. Let's spend some time talking about the implications, because they're actually huge, okay? This condition, polyploidy, having more than 2 complete sets of chromosomes, is abundant in the plant kingdom. And in fact, of the flowering plants, we expect about 50 to 80% of those species to have undergone a recent polyploid event. And it results in sort of this interesting cycle, where a plant will undergo a polyploid event, right, resulting sort of in a new species with 2 sets of chromosomes, or with multiple sets of chromosomes, like up to 4, right? And then it will undergo evolution, where those chromosomes will evolve. They'll basically undergo different kinds of chromosomal mutations, which we'll talk about a little bit more on Thursday, resulting in sort of a rediploidization, and then it happens again, polyploidization over and over again. So you get these repeated cycles of polyploids. Right? And what it means is that there are some plants that have really huge numbers of chromosomes in the cells. Okay? A diploid cell of an adder fern or adder fern has more than 1,000 chromosomes in every nucleus, right? Just because of repeat rounds of polyploidization. Okay? And as it turns out, polyploidization does have dosage effects in development that plants can survive that people can't. Right? Some of those dosage effects, right, are things that we select for in crop plants. I'm going to talk about those in a minute, right? One of the things that's sort of important for that is that we often want to produce plants anymore that are seedless, right? They produce seedless fruits, and that's so you don't have to spit them out. So when you look at a seedless fruit like a seedless orange, or every banana just about you've ever bought in America, right, there are no seeds in bananas, right? It's because they're triple. They've got 3 sets of chromosomes, so they can't do meiosis right. They never form a viable ovule, therefore they never form a viable seed, right? And you can eat these without having to spit everything out. However, just something I want to point out to you, 1 in every so many bananas undergoes that spontaneous doubling of chromosome numbers and suddenly will produce seeds, right? At one point in my life, I got really interested in this. And bananas, incidentally, are the cheapest of the seedless fruit, right? By far. Bananas are super cheap. It's the only reason I think people eat them, right? So I was eating a banana every day, and my hope was I'm going to get one of these spontaneous hexaploid bananas that's got 6 sets of chromosomes, so it produces seeds. And when I was a postdoc, it happened, right? And that was the last banana I ate. Right? I was like, I've achieved what I need to achieve. I ate this fruit, and I'm not eating it. Peak banana, yeah. I was at peak banana, and I couldn't get the seeds to grow though, which is maybe more on me and my lack of horticulture, because I really tried. But this is also really common in really important staple crops. So we're going to talk a little bit about wheat, which is an allohexaploid, which means not only does it have 6 sets of chromosomes, they're not all from the same species. Okay? Strawberries are an interesting case when My grandparents were young. Strawberries were all diploid, and that meant they were really small and kind of hairy, right? When about the 1950s, 1960s, they engineered a tetraploid strawberry. And so the fruit was bigger and juicier and more delicious. And now we've got these octoploid monsters that are the size of a baby's fist, right? Like they're almost like the size of like a peach or a plum, right? And they're better. They're just way better, right? And lots and lots of phenotypic effects of polyploid that largely result from this dosage effect in development. One is that they just generally have larger cells, right? And strangely enough, the cells that are the largest compared to diploid cells are the guard cells that exist outside of stomates on the underside of leaves. So a lot of times, if you want to tell whether a plant is polyploid or not, just look at the guard cells. Generally, they produce larger flowers that have more nectar. They also produce larger seeds. So when we talk about grass crops, right, what we need from grass crops, things like wheat, corn, rice, oats, barley, sorghum, millet, etc., right, we basically are eating the seeds of those things, right? And so if they can make them big, it's better. So almost all of our grass crops actually are polyploid to some extent, right? They also produce larger pollen. They also tend to have slower growth and development, and it's sort of interesting because a lot of times, and this will come down here, I'll just say it now, if you have a polyploid that wasn't annual, its diploid ancestor was annual, it'll become perennial, right? Which means that it will overwinter as rootstock or something like that. So that means that they tend to be also longer lived. Oftentimes, they also experience hybrid vigor, right? And that's basically something that results when you are fixed or heterozygosity, right? So if you've got two sort of different parents that come together, right, and you keep all of the genes from both of those parents, you're always going to be heterozygous, right? And that basically will give some advantage evolutionarily, right? One of the things that sometimes happens with polyploid plants is that they become weedy, right? Which means that they're able to grow more places than their diploid ancestors can, and so they become generalists, right? They can sort of occur in a lot of human disturbance. And there's some good evidence that humans have done a lot to help polyploid plants spread across the world. Now, this is actually evolutionarily important and has been even before humans were sort of helping to spread this. And that's because polyploidy results in instant reproductive isolation. You guys, has anybody here taken evolution, or do you have to take this before you take evolution? Take this before you take evolution. Really quickly, the cause of all species events, all speciation is caused by reproductive isolation. And that just means that you have two lineages in a single population that are no longer able to exchange alleles. They can't reproduce, okay? And so when you end up with something that's polyploid, right? So let's say that we've got a tetraploid that forms over here, right? It's a 4-end diploid cell. This will undergo meiosis to produce a 2-end gamete. And I'm going to show that it's a gamete by putting a tail on it. It's a plant gamete, so I'll put details on it. Right? Everything else in the population is 2-end, right? Right? And so these will undergo meiosis to produce 1-end gametes. Well, when these come together in syngamy, they make a zygote, which is 3-end. When it's got 3 sets of chromosomes, it can't undergo meiosis correctly, right? Because you don't have proper homologous pairing, so this is sterile. And so what that means is any sort of other trait that this evolves to keep it from mating with this is going to be selected for. And so you have instant reproductive isolation, right? Which results in really, really rapid speciation, right? So rapid, in fact, that there are many instances of us actually observing polyploid speciation in situ. We can see new species evolve, right? Which is kind of goes against what most people think about like evolution and speciation. Right? People think it's very gradual, it's very slow. This allows a really rapid way for speciation to actually take place, right? Another thing that's super interesting about that is a lot of times the polyploids are ecologically different from their diploid progenitors. We talked about all the ways that they're different. One of those ways is they become perennial and they can grow in a wider array of a habitat. And so that means that you have an ecological differentiation. One of my favorite examples of this, of these two things, is a well-known hybrid complex that occurs in one of the larger genera that's native to North America, which is Helianthus. Well, here's a fun thing about botany and politics. is that you guys have probably recently seen that the sunflower has been used largely as representative of Ukraine, right? And that's because Ukraine is a really largely agrarian country that grows tons and tons of sunflowers, okay? They're not Ukrainian. They're North American. It's a North American plant, right? But it just got moved over. Plants move all over the place all the time. When I was in China not that long ago, right before the whole COVID thing, I was staying in southern China near the border of Laos, and I met some people who were from Thailand, and they were talking about frangipani, which is the national flower of Thailand, which is Mexican in origin, right? But it just grows really well there, right? And people sort of adopted it. So it's sort of the same thing, but this is one of ours. This is a native to North America. Species that most people are familiar with is Helianthus annuus. And this is a species that we grow as a crop plant, right? And that's because it produces these enormous flowering heads that produce lots and lots of fruits, right? Incidentally, there are lots of flowers here. This isn't one flower. That's a botany lecture that I'm not going to give right now, right? This is an inflorescence, but it produces all these fruits. We call them sunflower seeds, right? We eat them, but we also get oil from them. It's a highly productive crop, especially when you go right to the middle of the country, Kansas, like just fields and fields, okay? On the other hand, there's a really common Helianthus species called Petiolaris that grows pretty much all over the United States, mostly in the western United States, where Helianthus annuus' native range is probably in the east. Well, what happened was, when people started cultivating Helianthus annuus and planting it throughout the Great Plains, it came into contact with Helianthus petiolaris, right? And these things were actually able to hybridize, right, to create this polyploid hybrid species, which is called Helianthus paradoxus. Now, this has traits of both parents, but it doesn't like either of the environments that its parents live in. This actually is a plant that's really well adapted to highly saline soils of the Great Basin area of Utah and Nevada. So completely different ecologically, can't sort of easily backcross with its parents, brand new species. Another important thing to keep in mind is that polyploidy results in an increase in genetic variation over the diploid progenitors, and that's because a polyploid, of course, has all of the alleles of its parents. It doesn't have half, right? And so you can end up with fixed heterozygosity, resulting in some of that hybrid thing. Any questions about this? So how does this actually occur? Right? Well, we've already spent some time talking about how aneuploidy occurs. Not a surprise, this happens in much the same way. You get non-disjunction, but it's a much more systemic non-disjunction, usually from the failure of the centrosome to form, right, in meiosis, so that all of the chromosomes end up in just one cell rather than being divided among two. It can happen in meiosis 1 or meiosis 2. And so when you have two, the other thing is we know how to do this experimentally. This is sort of fun. There's a toxin that's produced by a species of lily called colchicine, and that prevents spindle fibers from forming. And so if we want to create polyploid plants, we basically will just treat anthers with colchicine, and you get a bunch of diploid gametes that form from that. When we have those come together into one zygote, right? So we're combining, gametes of different ploidies into one zygote, right? We call chromosomes that have a similar arrangement of loci that came from different parents homologous, or they're not homologous, it's like homologous or something. Homologous? Homologous. It's just an extra syllable in there. Just put an extra syllable in there. And so that basically means that they can pair, but sometimes with really reduced affinity. So they don't pair necessarily great all the time. And it isn't until you end up with some chromosomal mutation that ends up sort of swapping them out so you get better pairing. So oftentimes you start out with a polyploid that is not especially fertile, but it will get more fertile over time with its ability to self. Okay? We divide polyploidy into really two classes. One, which is called autopolyploidy, which is multiplication of a single chromosomal set. This usually happens in a parent that undergoes polyploidization of its gametes and then cells. Right? So there are no new chromosomes coming together. It's just multiple sets of the same old chromosome. And then we have something which is called amphipolyploidy, and that's merger of different sort of chromosomal sets. Right? So the parents have some chromosomal differences that are contributing to this. And of amphipoid things, if the species are from completely different parents, like different species, pardon me, the parents are of completely different species, then we call that allopolyploid. So that's what Karpachenko was doing. He was making these allopolyploid Raphana brassica species. Make sense to everybody? Talk about each one of these at a time because they're both sort of important and interesting. Autopolyploidy is surprisingly more common but less known of the polyploids, and that's simply because you don't end up, by putting any new sets of chromosomes together, you don't end up with any new allelic combinations. Mostly results in changes in dosage effects. Okay? But this happens when you have spontaneous doubling of chromosome number, right? So you can have a diploid adult plant, which we call sporophyte. Don't worry about that. This will undergo meiosis, but if meiosis fails, right, because you have a failure of spindle formation, you can end up with diploid gametes. And when these diploid gametes come together in self-fertilization, which is really common in plants, right? Most plants have flowers that have both male and female sexual function, so they can basically self-fertilize. You end up producing offspring, which are tetraploids. So they've got 4 sets of chromosomes rather than 2, and of course that's more than the parents have, and they are reproductively isolated from the parents. So in some ways this is instant speciation, right? Because they can't share a gene pool with their parents, and now they're evolutionarily independent, right? And this is something that sort of happens all the time, and in some plants happens recurrently, right? So that you can find populations of diploids that are constantly producing tetraploids. But because you're not actually taking alleles that have never been together and putting them together, this doesn't result in a great increase in diversity. Rather, what it results in is the production of plants which are pretty similar to their diploid progenitors, but maybe different only in those things that we listed, right? So they've got maybe larger flowers and fruits, flowers that have more nectar and things like that. But as you can imagine, this is super important for agriculture, right? Because inducing polyploidy, autopolyploidy, is really, really easy. If you've got a plant that's capable of selfing, just treat the flowers with colchicine, let it self, and then screen the offspring for polyploids, which will produce bigger fruits, right? Bigger flowers, all the kinds of things that we want in our crop plants. Allopolyploidy is a little bit weirder and a little bit different, but also we're going to find out is important agriculturally. Allopolyploidy is much more like what Karpachenko discovered, right? Because when you have 2 species that are different, they're different species, and they produce offspring that are sterile that will then undergo a spontaneous doubling to produce fertile offspring. We have a great example of that that took place in the harbors around southern England, London and things like that, where there was a species that always occurred there, Spartina marittima. It's a seagrass that's got a diploid number of 60 chromosomes, right? So 2n equals 60. And as ships were leaving England and sailing into Boston Harbor, they picked up seeds from a species that occurred there that was closely related, Spartina alterniflora, which had a chromosome number of 2n equals 62. And so the seeds would stick to the boats, they would come back and would work both ways, right? You have like sort of this exchange of seeds, and so we had these mixed populations of 2 different species, Spartina alterniflora and Spartina marittima. And what happened in both places that you started to have these sterile hybrids form, right? They had some of the traits of one, some of the traits of the other, but they didn't really make seeds. They never really set seed until they did, okay? And what they discovered was that the sterile hybrids, of course, had 30 chromosomes from marittima, had 31 chromosomes from alterniflora, and so had a diploid number of 30 plus 31. Doesn't really work out in meiosis. But as soon as they underwent a spontaneous doubling, essentially an autopolyploid event, right? They went from 30 plus 30 plus 31 plus 31. And this is a new species. It's got some new traits, Spartina anglica, which has a diploid number of 120-21 chromosomes. Okay? And so that's something that happened in human history. It's a new species that arose that was witnessed and documented as an action. There's a lot of cases of things like this. This is important in agriculture because we've taken a lot of time to try to deconstruct the story of one of the most important crop grasses, which is wheat, right? I think probably 1/4 of the calories that people get on the planet come from this one plant, which originated in the Tigris and Euphrates valleys of the Middle East, what they call Mesopotamia, probably around 11,000 years ago. And people at the time were sort of new agrarians, right? They were sort of changing from a nomadic herding lifestyle to a stay in one place and grow food lifestyle. And they were probably growing a lot of this one species, which is Triticum monococcum, right? And we're going to basically represent its genome, its diploid genome, with the letter A. So this has got 2 copies of that genome, so it's AA and diploids, okay? We know at some point in the past that this hybridized with an unknown species of Triticum that exists, it's somewhere but doesn't exist anymore as far as we know. But we still have that genome because we have its descendants. So this probably made a sterile hybrid that had some of this B, some of this A to make AB. Now these don't match each other, so this can't undergo meiosis. Unless what happens? What would have to happen for this to be able to undergo meiosis? Spontaneous doubling, right? So we get AABB. Now every chromosome has got a buddy, right, in homologous pairing. And that results in something that's called emerald, which we know that people have been using for at least 10,000, almost 11,000 years. And this is Triticum turgidum, and it's got a diploid number of 28. There's another thing that occurs in the Middle East, which is called Triticum tauschi. This still exists, so we're able to sort of sequence it and know what this genome looks like. And this basically supplied a haploid gamete to the diploid gamete produced by T. turgidum to make a triploid sterile hybrid, which has an A, a B, and a D, right? So this has got a triploid number of 21. A, B, and D is, of course, going to be sterile until it undergoes what? Spontaneous doubling, right? And so that we end up with an AABBDD genome, and this gives us Triticum estevan about 9,000 years ago, which is what we know as bread wheat today, right? So this is the wheat that is in my sandwich, which is in my bag right now. That's sort of bread wheat today. Really, polyploidy is incredibly important for the development of it. And when we think about it, right, some of what we know about polyploidy makes sense, right? We look at Triticum monococcum, right? It's got really, really small seeds compared to its polyploid descendant, right? Which has got really, really massive seeds, right? These are much more vigorous plants. Right, so yeah, you can grow wheat in the Middle East, but you can also grow wheat in Canada, right? It grows in some places that, you know, its ancestors didn't grow in, right? Sort of a huge success story of the Middle East, and ultimately, when you think about it, this is sort of how civilization happened. People can't really have a civilization until there's enough calories to go around. And as soon as there are enough calories to go around, then everyone's job doesn't have to be making calories. People can do other things, like, you know, learn how to smelt ore and I don't know, invent writing and wheels and aqueducts and shit like that. Sort of one of the major civilizations depended on its domestication. I think that's all I'm going to talk about today, which is because I don't have that much time. We're going to move into sort of how major changes in chromosomal structure take place on Thursday. Does anybody have any questions about what we covered today? No? All right, I'll see y'all on Thursday.